And I'm so thrilled now to welcome Dr. Paul Schreiner to the show. Paul is a fellow Penn State alum. We are Penn State, right? And he recently retired from the USDA, where he worked as a research plant physiologist in the horticulture crops production and genetics improvement research unit in Corvallis, Oregon. He specializes in research on grapevine physiology with a focus on plant nutrition and the functioning of roots and arbuscular mycorrhizal fungi. So we're going to talk about that today quite a bit. So, hey, Paul, thanks for taking the time to join me today for a conversation.
Thank you for having me, Fritz. I'm glad to be here, and hopefully we'll have a fun conversation about some of your friends below ground.
Yeah. Well, this is the Vineyard Underground podcast. So this is really our chance to dig underground and talk about what's going on down there, because most people can't see that with their eyes. And it's something that I think needs to be explained. I think there's a lot of visual that could go with this. So our job today is to explain mycorrhizal fungi without visuals. And I know we can do it. So, but what I'd love to do first, Paul, is, get to know you a little bit and introduce you and your background. So I'm curious how you got interested in plant physiology and specifically focused on grapevines and mycorrhizal fungi in the vineyard and just nutrition in general. So how'd you get to where you are?
I'm not a typical scientist case, maybe, because I didn't have a plan on what I was going to do. I went to college. In fact, when I graduated high school, they were like, "Oh, you're good at math, so be an engineer." So I didn't know what an engineer was. So that's where I started, and then I'm like, okay, this is not for me.
Yeah.
And moved into biology, and then it just kind of went in that direction. And then, at Penn State, I got a BS in biochem. And then I realized after I got the BS, I can't really do anything with this biochem degree except go to grad school. So that's what I did. And, I didn't really want to work on human beings or animals. So plants and understanding plant biochemistry seemed like a good opportunity. And so that's kind of how I ended up being in plants.
I want to start off by just explaining a little bit about mycorrhizal fungi. What are they? Why are they important to vineyards? There's different types. So maybe we can just start with that.
Okay. Yeah. So mycorrhizal fungi are grouped into about 6 different types. Okay. The 2 main ones that most people know about are called ectomycorrhizae and then vesicular arbuscular mycorrhizae. That's the old name, VAM, right?
Yeah, I remember when I was in school, it was VAM, and now it's just AM, right? So what happened?
So the deal with that is early on, these fungi, the vesicular arbuscular type, made both vesicles in roots and arbuscles in roots. But as they learned more, certain species or genera of the fungi do not form vesicles. So they don't all form vesicles. So then the name became just arbuscular mycorrhizal fungi. So it went from VAM to AM. But I think AMF is maybe a better acronym, arbuscular mycorrhizal fungi.
AMF, got it.
And so the AMF fungi probably helped the first land plants actually establish on the land, on soil. It's pretty interesting. There's 400 million year old fossils of very, very early land plant roots that have been cleared and stained, and believe it or not, they have arbuscules in them.
Oh, wow.
So yeah, I mean, it's amazing that it was even possible to see that in a fossil, but yeah, so I mean, they've been around since the dinosaurs. And in fact, if you look at the evolution of this group of fungi, based on molecular clock, which is a different approach, right? It's, the accumulation of mutations. They probably date back 1 billion years when they really evolved from other fungi. So they're very old. They've been associated with plants ever since plants have come onto the land. And so there's, it's a very interesting situation. And basically they act as an extension of the root system and help plants obtain nutrients from soil, and probably water in some cases. That's less clear. But the AMF fungi are different than the ectomycorrhizal fungi. So the ectomycorrhizal fungi are much more common, particularly in forest trees, conifers, and a lot of the ectomycorrhizal fungi evolved much later than the AMF fungi. And they're in completely different classes of the fungal guilds, based on the tree of life.
Good old Dr. Jonathan Lynch was my plant nutrition professor there. Blew my mind about the world of nutrition. He talked about mycorrhizal fungi. And I remember distinctly in his class, he mentioned that there's been studies showing if you apply phosphorus, it can actually be detrimental to the associations of these fungi. So it does beg the question, if we already have ample phosphorus in the soil, but more importantly, if we do plant tissue analysis and we show that the phosphorus levels are sufficient in the plant, is it a bad idea to add a little phosphorus to the soil just in case? Or is it going to actually hurt our mycorrhizal associations? Or if people put out like a triple 13 fertilizer and they really only need the nitrogen and potassium, but they're throwing phosphorus out there too, does that disrupt this relationship?
If there's a lot of phosphorus in the plant, or if there's a lot of phosphorus in the soil, the mycorrhizal fungi are not so necessary, right? And so, you could ask the question, can plants survive without mycorrhizal fungi? Yes, they can, especially if you give them a bunch of phosphorus.
Yeah.
Okay. But the plant itself does have some control, right? And so if you have a lot of phosphorus, the plant is sending signals that say, I'm phosphorus sufficient. And so those signals that are going down the root system are telling those roots, we don't need phosphorus. So we're going to put less sugar down here. Less sugar means we're going to turn off mycorrhizal fungi to some degree. But there's also much more definitive regulation. There's, in fact, in most plants, there's a specific phosphate transporter, at least one, probably more than one, that is specific to mycorrhizal fungi, so that when mycorrhizal fungi colonize the root, the phosphorus goes through this particular transporter. And if you knock that transporter out, like making mutant plant, it can no longer use that transporter for phosphorus that it uses for mycorrhizal fungi. Colonization stops. So the plant has some control. The question is, how much control does the fungus have? And that part we don't quite know yet. But the fungus probably has some control as well. And so that's where this whole arbuscule thing comes in. So the arbuscule, we know, is the site of exchange between the plant and the fungus. So that's where carbon is going from the plant to the fungus. And let's just, for now, talk about phosphorus. Phosphorus is going from the fungus to the plant. And so that happens at this arbuscule, which is this highly branched structure. It obviously takes an immense amount of energy to make this, because both organisms are building all these membranes and cells and all, not cells, but membrane around this tree-like structure to accommodate each other. So a lot of energy is happening there. And so if you knock out the phosphate transporter, the arbuscule no longer develops, or, they won't develop anymore. And then the fungus can't get carbon, and the plant is not getting phosphorus through that, through the mycorrhizal channel. So most plants also have other phosphate transporters that are on the epidermis of the root cells that are trying to get phosphorus just out by itself, like not using the mycorrhizal channel.
Can you test your soil or your plant roots? Or if you don't have grapevines yet, can you test the native plants there, the grasses, trees, and other things that form these associations? Anyone doing anything like that? Or is it possible? Or maybe it's possible, but no one does it because it's too costly or just not common?
Well, yeah, I think the bottom line is it's very costly to do. It's a very technical thing. I mean, it's like other lab analysis things. I mean, it's technical, but there's just not a lot of people doing it. And, there was a company here in Oregon that was doing that for people, doing assessments. in order to know if you have mycorrhizal fungi, you basically have to dig up roots and clear them and stain them, and then that reveals the mycorrhizal fungi inside. So it requires a bit of equipment, and not a lot of people do this. And I think part of that is there's not great demand. So can you get it tested? Yes. You might have difficulty finding anyone in your area to do that. And for the most part, it has been more of a thing done by scientists.